Welcome back again to a new video tutorial session from Biology Boost Up. Today in this video tutorial session we going to study about the mechanism of gene regulation and the Lac Operon of the prokaryotic cell. So now let's start. At first we going to study what is operon, a group of genes making up a regulatory or control unit. The unit includes operator, promoter and structural gene. Now next, what do you mean by inducible and repressible system? The enzyme whose full level of transcription does not occur unless their effector molecule are present is called the inducible enzyme. Such effector is called inducer and such regulation is called inducible system. For example, synthesis of beta-galactosidase enzyme is induced by lactose. Here the lactose acts as a inducer. Now next, the enzyme whose full level of transcription occurs until such time as their effector reach a critical concentration at which transcription is inhibited. Such effector is called co-repressor and such regulation is called repressible system. For example, tryptophan synthesis. In absence of tryptophan, the enzymes are synthesized at full level. Now from here we can conclude that inducible system are off until the effector is present and repressible system are on until the effector is present. Now let's talk about our main topic of this series is Lac Operon. The Lac Operon of E. coli contains genes involved in lactose metabolism. It's expressed only when lactose is present and glucose is absent. Now next point, two regulators turn the operon on and off in response to lactose and glucose level. The Lac repressor and catabolite activator protein, that is CAP. Next, the Lac repressor acts as a lactose sensor. It normally blocks transcription of operon but stops acting as a repressor when lactose is present. The Lac repressor senses lactose indirectly through its isomer allolactose. Now coming to the next point, the catabolite activator protein or CAP acts as a glucose sensor. It activates transcription of the operon but only when glucose level are low. Now the CAP sends glucose indirectly through the hunger signal molecule, that is CAMP. Now next, what makes the Lac Operon turn on. In E. coli bacteria, they can break down lactose, but it not their favorite fuel. If glucose is around, they would much rather use that. Glucose requires few steps and less energy to break down than the lactose. However, if lactose is the only sugar available, the E. coli will go right ahead and use it as an energy source. To use the lactose, the bacteria must express the Lac Operon genes, which encode key enzyme for lactose uptake and metabolism. To be as efficient as possible, E. coli should express the Lac Operon only when two conditions are met. The first one is lactose is available, and the second condition is the glucose is not available. Now the structure of Lac Operon. The Lac Operon contains three genes, that is Lac Z gene, Lac Y gene and Lac A gene. Those genes are transcribed as a single mRNA under control of one promoter. The lactose operon, also known as the Lac Operon, is a set of genes that are specified for uptake and metabolism of lactose and is found in E. coli and other bacteria. The Lac Operon consists of three structural genes. The Lac Z genes, which codes for beta-galactosidase, which acts to cleave the lactose into galactose and glucose. The Lac Y gene, which codes for permease, which is transmembrane protein necessary for lactose uptake, and the Lac A gene, which codes for transacetylase that transfer an acetyl group from coenzyme A or CoA to the hydroxyl group of galactosidase. In the 5' end, with respect to Lac Z, is the Lac gene, which encodes a repressor of the Lac Operon, which is transcribed independently from the structural gene. In addition to the three genes, the Lac Operon also contains a number of regulatory DNA sequences. Those are regions of DNA to which particular regulatory proteins can bind, controlling transcription of the operon system. As you can see here, the promoter is the binding site of RNA polymerase. This RNA polymerase helps to synthesize of new mRNAs from the functional genes, that is Lac Z gene, Lac Y gene and Lac A genes. Now, the operator is a negative regulatory site bound by the Lac repressor protein. The operator overlaps with the promoter, and when Lac repressor is bound, the RNA polymerase cannot bind to the promoter and start the transcription. Now, The CAP binding site is a positive regulatory site that bound by catabolite activator protein or CAP. When CAP is bound to this site, it promotes the transcription by helping the RNA polymerase bind to the promoter. Now the next part of this video series will be coming very soon, and thanks for watching my video. Please like, share and subscribe my channel. Thank you.